Yeah, hello everyone. I'm Xiaowei, a PhD student at Ji-Xin Cheng's lab. It's my honor to present our work on cancer and microbiome. Next slide, please. Uh, the metabolism involved in cancer development, drug interactions, and the relation between cancer and other communities in our body are the key to better understanding, hence better diagnosis and treatment. Precise and multimodal characterizations of intracellular and extracellular features are essential to depict the full view. Our lab focuses on exploring the endogenous molecule spectral features across range from visible to mid-infrared to enable a label-free detection in biosamples, thereby remaining the sample condition undisturbed. So by applying visible femtosecond laser, transient absorption is a major tool for identifying and quantifying colorful micromolecules. At near-infrared range, coherent Raman scattering, which provides bound vibration information equivalent to spontaneous Raman, but provides million times faster emitting speed as the vibration is boosted by two ultra-fast near-infrared lasers. From 1.2 to 1.8 micrometer could pump the molecule to overtone vibration mode, which will convert into ultrasound waves. This method enables deep penetration depths and detection of cancer margin. So at the mid-infrared range, our lab combines the visible with mid-infrared to overcome the IR microscopy intrinsic low resolution problem while maintaining the large absorption cross-section in IR. So next slide, please. In our previous study, by combining the SRS, which can map the lipid droplet inside the prostate cancer cells, and spontaneous Raman, which analyzes the fingerprint of the lipid droplet, and aberrant cholesterol ester accumulation is found. And by depleting cholesterol ester, cancer proliferation is largely inhibited. In the following study, more metabolic markers were investigated for the cancer cell. By comparing the lipid droplets in CH region, the unsaturation level can be quantified by hyperspectrum SRS. The high unsaturated level of the lipids were found in ovarian cancer stem cells, and the lipid desaturase inhibitors can inactivate the pathway that regulates the expression of lipid desaturases and the ovarian cancer stem cell stimulus. These researches shows that the ability using single cell chemical mapping for metabolism characterization and the potential for exploring more metabolism markers and the pathways of cancers for better diagnosis, and a step further help develop a new treatment method. Next slide, please. Beside the cancer cells, our lab is actively working on the microbiome field due to its increasingly importance in human health. In our recent work, by combining two regimes, so that's SRS and fish fluorescence in situ hybridization, we have some interesting findings in human gut microbiome. So deuterium water is used in culture media in order to deriving deuterium into biomass. In the cell silent region, the CD peak can be used as a quantification target, offering metabolism activeness information. And the fish selectively labeled bacteria based on their genetic information with fluorescent signals, offering identity information. So next slide, please. On the left top, there is our SRS fish platform, which combines the femtosecond SRS with two photon fluorescence. The mucus, so at the middle, there is an illustration of our experiment. The mucus layer is the main nutrient source for the gut microbiome, but which part of it is the essential carbon source is still a question. So in our study, by adding different mucosal sugars with 50% deuterium water to create various growth conditions, the bacteria in the gut are labeled to different extent due to their favor of different nutrient source. So fish label two main groups in the gut microbiome, which help us to visualize the target we are interested in. And on the bottom, on the top right, we can see there is a fish images, and we use the fish images to create the fluorescence measuring mask. And from the SRS result, we have the metabolism activeness. And on the bottom, we can see that's the labeling efficiency of different microbiomes inside the gut microbiome. And using different nutrients, we can see their response are actually different. And for potential applications, we can use SRS fish this platform to research on the human gut microbiome metabolism response to different cancer therapies, and we can observe the genotype and phenotype information simultaneously in situ and remain the sample undisturbed. And that's it, thanks.